Welcome to Scigest, your fortnightly serving of digestible science from plant and food research.
Welcome to Scigest. Today you'll be joining me to learn about fish cell culture from Georgina Dowd. Originally from Ireland, Georgina did her PhD at University College Cork, where she studied host pathogen interactions using human cell models. Following her PhD, she moved to the University of Otago, where she continued her work looking at the molecular mechanisms of infectious disease in humans. After joining plant and food research, Georgina established New Zealand's first dedicated fish cell biology lab, where her team now work on developing cell lines from finfish that are relevant to New Zealand. They develop these cell lines for several applications that support aquaculture, but also use them for novel applications such as cellular agriculture. So, thanks for joining me today, Georgina, and welcome to Scigest.
Thank you for having me, Julie.
I guess the first thing that maybe we can cover is if you can tell me a little bit about your research area and what's led you to what you're working on nowadays.
Yeah, sure. So I joined plant and food research in 2016, I think. Yeah, 2016. And when I got here, I had come from a very fundamental background. And coming across into the applied world was a big change for me. And my boss at the time, Susan Marshall, came to me and said, "Can you culture cells from fish?" And I said, "Sure, no problem. Shouldn't be too challenging." How naive I was. But that led me on this amazing journey to where I am right now, which is leading a team of scientists and researchers looking at fish cell biology from New Zealand relevant finfish. So in general, fish cell biology is hugely unexplored. Only 2% of the world's fish species are represented by cell lines. The technology originated back in the 1960s to make cell lines for looking at viruses that were decimating aquaculture species. And it didn't progress much beyond those applications in the years that followed. And it's only really in the last 10, 15 years that the field has really started to advance.
Okay, I think we've all been there with how hard can it be?
Yes.
Yeah.
That optimistic naivety.
Yeah. What were the first fish species that had cells cultured? Out of curiosity.
Good question. So actually there was a Chinook salmon embryo cell line that was made in the States. There's some bluegill. There's a lot of rainbow trout cell lines. Rainbow trout is probably the most represented species in fish cell lines currently. There's been a lot of research done on cell lines from that species.
And you mentioned in that introduction that fish cell lines were originally established to help isolate viruses that were causing problems in aquaculture. Mm-hmm. Are we still doing that? Yep.
So it's still one of the main uses of fish cells in research today, but we also use them as part of global surveillance systems for notifiable diseases, and that's with the World Organization of Animal Health. They have a set of standards for looking at viruses and monitoring viruses that are potentially lethal, particularly for aquaculture industries around the world. So obviously viruses need a host cell to replicate and survive. So we can use fish cell lines to both isolate and propagate viruses. And that's really useful then for understanding those viruses and potentially how to treat for those viruses through vaccinations or therapeutics.
Okay. Yeah. So fish deal with pandemics just like people do?
Exactly. Exactly like them. So it's always a big concern for industry, for the aquaculture industry globally, that, something like COVID-19 may come along and we're not prepared. But with having cell lines, we can be a lot more agile in the response for such incursions that might happen. Yeah,
that makes a lot of sense.
Mm-hmm.
Are there any other types of diseases that you can explore using cell culture approaches?
Yeah, so essentially any disease-causing agent, we can use cell lines to look at that. So bacterial diseases, fungal diseases, and we can use those cells to look at, get into the nitty-gritty of what's happening at a cellular, molecular level between the pathogen and the host. And like I said, use that knowledge and use the cell lines then for looking at vaccine development and therapeutics. So they've played quite an important role and will continue to play quite an important role in fish health in terms of infectious disease.
So on the whole, it sounds like this sort of disease research can help us keep our fish happier. Can you go into any more detail there?
Yeah, so I mean, there's lots of other ways we can use the fish cell lines to, I guess, help maintain the health and wellness of our fish. So obviously the fish's environment is very, very different to our own. They're in an aquatic environment, so their exposure to potential toxins or infectious agents in the water is quite different to our own. So we can use cell lines to understand their responses. We use the cell lines as a model for their responses in the aquatic environment to toxins or toxicants in the water. These would include things like microplastics, you might have agrochemicals, pesticides, insecticides, that kind of thing, heavy metals, oils and oil derivatives. So we can use the cell lines as models to look at how a whole fish might actually respond to these toxins in the water. And actually in the last couple of years, in 2021 I think it was, there was a huge step forward for fish cell culture globally when we had a new standard validated by the OECD, and it's called the fish acute toxicity test using a cell line. So usually when a new chemical is created, they undergo toxicology testing as part of the regulatory landscape. They have to determine the LC50 and how toxic it will be to the aquatic environment, and they use whole fish for this. Quite a stressful analysis to be done both for the animals, for the researchers as well, I guess. So we now have a validated alternative method to that, and that's using fish cells from rainbow trout. So it's really fantastic to see that moving forward, that we can now start using validated methods to replace those whole animal analysis studies.
Yeah, that does sound a lot easier and a bit more ethical as well, where you don't have to stress out the fish just to learn what's safe for the environment.
Exactly.
Yeah.
And it's a lot more high throughput as well, so we can do it a lot quicker. So obviously industry quite likes that as well.
Yeah. Is it lower cost to have a cell culture versus a fish? Because I imagine that it's not necessarily all that cheap, right?
No, I mean, it's way cheaper to do these analyses in a cell model than it is to do in a whole animal model. Of course, if you do a whole animal model and you're looking at varying one condition, in this case looking at the impact of a chemical, you would need to maintain every single other condition, so the temperature, the dissolved oxygen in the water, you need to maintain every single other thing, which can become very, very expensive. And of course you need to maintain the fish the whole way up until you do the actual experiment, so it ends up being very, very costly for companies. So these alternative models are significantly cheaper to do. Yeah.
Okay. And you mentioned that this new standard is in rainbow trout cell lines.
Yes.
So the results from that, does that also give you good information on other fish species?
Yes, it does. And I mean, there's been a lot of work done on validating that method and comparing it to the killifish, which is one of the whole fish that's used in those toxicology tests. And it's quite comparable. With that said, we do see in the lab, we work with two species, and we can pick up differences in the response between those two species at a cellular level. Quite a clear example is when it comes to temperature. One of our fish is a cold water fish. We can keep those cells in the fridge quite happily. One of our other fish is a warm water fish. If we put those cells in the fridge, they will die within, 12 hours, they're gone. So we can see differences across species, and that's something that I'm particularly interested in is really unraveling how different species react at a cellular level.
So I guess some species might be a little bit more sensitive to some of these toxins or environmental agents than others, yeah.
And that becomes quite important then for species, we'll say, that are endangered, or, our native species, we want to protect those as much as we can. So the more information we know about those, the better. And what's really great about doing fish cell culture is we can create cell lines in a non-lethal way. So we can take a small portion of a fin clip and carry out a lot of analysis on that, and the fish will stay alive. So it's quite a unique way of investigating a lot of different things without doing any damage at all.
Can you get different tissue types
Yep.
cultured from the fin clip, or does it tend to be pretty set already by that point?
I would say in general, we find that the fish cells are very plastic. They seem to adjust to the conditions that they're in very quickly and in most cases quite happily. Usually if we wanted to, say, isolate an epithelial cell from the gill, we would get a gill cell rather than trying to manipulate a stem cell to produce or to differentiate into a very particular cell type. And that's usually the way it's been done in fish cell culture. There hasn't been a whole heap of cell sorting done. So dividing, when you get a big mix of cells out, that you can separate them out into specific types of cells. It's something that we'd love to be able to do, but for now we just go to the specific tissue type to get cells from that. So if we wanted gut cells, we would go to the gut.
Yeah, that makes sense. But I guess if you had like a very protected species or something, then maybe going that stem cell route would make sense in that case.
For sure.
We've covered quite a bit of ground now in terms of ways that fish cells and cell culture are used to support traditional aquaculture and fish health. But I think you also work on a few more novel technologies using fish cells. And I heard you recently got some funding in this area, so congrats on that. And do you want to tell us a little bit more there?
Sure. So while a lot of our research does focus on supporting fish health and wellness in traditional aquaculture settings, we're also looking at using our fish cells in really novel ways. We call them fish cells as production systems. The technology is kind of more widely known as cellular agriculture, and that's essentially the production of agricultural products from cell lines. So people call them lab-grown burgers, cultured meat, in vitro meat. There's lots of different names for it. But really, we like to think of ourselves as production systems where we can use them to make things for us. So, for example, we might use them to make extracellular matrix components like collagen, or use them as a production system to make fish meat or fish protein, and that can be for animal or human consumption. So we recently got our MBIE Endeavour research program to pursue this work. But really the work that we want to do in that program, it doesn't just help cellular agriculture, it actually helps all of the things that we've just discussed, the virology, the toxicology, all of those factors that we can use cell lines to help with traditional aquaculture and fish health, we can also use with this new technology. One of the big challenges of cellular agriculture in the seafood space is that huge dearth of information that I mentioned earlier on. The fundamental research just hasn't been done. So while the cultured meat sector may be further ahead, we're still kind of struggling a little bit with the fundamental research. And I think the sector as a whole maybe were like me at the very start, naively thinking, yeah, we can do this. It shouldn't be a problem. It should be easy peasy. But there are still a lot of technical challenges. And probably for us, the biggest one that we see is that the cells in the media are the bricks and mortar of cellular agriculture. If we don't have those right, the sector is going to fail. The technology is going to fail. They have to be perfect. And for fish cell culture, since the 1960s, since that first cell line was developed, we've been using media and conditions that have really been optimized for mammalian cell culture. So our bid is really about understanding fish cell culture, what they actually need, looking at it in this fish-centric way, looking at the whole organism, seeing what it needs, and then applying that to our cell culture, and then using that to really develop that fundamental knowledge and capability to propel the cellular agriculture sector forward.
So it sounds like a really big overlap then between fish physiology and cell biology itself. Yep. So it's not one or the other.
Yeah, exactly. And I mean, I think, the more we can understand how the fish develops over time. We did quite a big study with a PhD student, Hayley Stent, looking at how fish muscle actually develops. And we looked at 2 different species, and we could see very clearly that they use different modalities of making muscle in vivo. So having all that background knowledge is really important, but also understanding what the fish actually eat is really important. how they use that feed conversion, how they use their food, what's in the food that they actually want. That's also really important for our research in the cell world.
I guess a big question I've got here is, how do you go from your fish to the cells in the dish? What are the main steps that you have to do there?
So we usually start with a piece of muscle from the fish. So that would just be like, a piece of fish fillet. So we only take a very small amount. We break apart those cells and we isolate them. We want to give those cells both the nutritional and environmental conditions to be as happy outside the fish as they were inside the fish. And if we can do that, they will continually replicate over years, and they will do so quite happily. Then we want to be able to give them the cues that they would get in the fish, outside the fish, give them those cues to form muscle. We will probably give them a scaffold to do that on. So that might be a plant-based scaffold, could be a 3D printed scaffold, and the cells will grow on that, form the muscle fibers, and that's essentially how you get that chunk of meat. Of course, in some cases, like an oily fish like salmon, you would need to introduce maybe some fat cells in there to give it that, to replicate or to mimic the fish meat in vivo. It's a very complex process to get a piece of fish meat together, and it's not really that well understood in seafood yet, I would say.
Okay. Yeah, because you mentioned salmon, and I'm vaguely aware of a company that's trying to culture salmon meat for sushi, I think, but it looks really complicated.
Yeah, and it definitely is. And I mean, in fairness to the field, and there are a couple of companies out there that have absolutely incredible prototype products, and it's worth a quick Google to see these because they look phenomenal. The big challenge there is scaling that up. Because like I mentioned earlier on, if we don't have those bricks and mortar right, scaling up those culture of those cells to such a degree that we're going to need for this technology to be a success is going to be very, very difficult.
Yeah, it sounds like a few challenges in the way there.
Just one or two.
Yeah. Are you in contact with any of these other companies or planning to work with them? Yeah,
I mean, we have a couple of stakeholders on our research proposal that we keep in touch with regularly. And, since the news of the grant has been announced, we've had quite a lot of people reach out internationally to be kept up to date with our work, which is great to see, because I think originally maybe the industry wasn't so keen on sharing information because it was like this race to market. Whereas now I think people realise actually there's so many challenges that we have to start working together. So it's a great opportunity for us here in New Zealand to start developing technologies and using the global market to get New Zealand out there. So yeah, we're really excited about it.
Yeah, it sounds really cool. Is there a specific species that you're targeting first for trying to grow a fillet in the lab?
We're looking to Chinook salmon.
Okay.
So it's, of course, New Zealand's only aquaculture finfish species. It's quite a lot of knowledge around that species, and there's quite a big demand for it on the global market. So that's where we're focusing our efforts right now. But we're hoping that with the technology we develop as part of the research grant, that we can actually apply the technology across several different species, and it will speed up the whole process a lot better than it is right now, I guess.
Yeah. So you'd be looking at a few different tissue types there with the Chinook salmon to make a nice fillet?
Well, for the Chinook salmon, we're just going to look at muscle.
Okay.
We're just going to look at muscle for right now. Yeah.
And is that also where you would start for the other species?
Yeah. If we're going to make cultured meat, we would definitely go there to the muscle. But obviously for different applications, we would go to different tissues. But for the fish meat, that's where we're going to go. Yeah.
Yeah. And is that because muscle is also really easy to work with, or are there other tissue types that are easier?
We want a very specific cell type out of the muscle. So we want those stem cells that are going to differentiate into muscle, and that's where they reside, is in the muscle tissue. So that's why we go there.
Do you have a favorite cell type to work with?
Yes. Maybe I have a favorite cell line that we've established, and that's probably one we developed from snapper mussel. I have a love-hate relationship with the cell line, because for a long time we couldn't figure out what it was doing. But then when we did figure it out and we identified what kind of cell it was, that was really exciting. And just being able to pinpoint exactly what it does and how we can use it was really, really exciting for us. So that's probably my favourite cell line, but I also love all of the cell lines from the gill as well, because they're very beautiful. They look really nice.
Oh,
so they're
cool.
they're
You'll
probably
have to
my
show
favourites.
me sometime. I want to see that.
They're really nice. I like them a lot.
Yeah. Well, we've covered quite a few, I guess, technical and biological challenges here, but do you see any other main challenges with using cell-cultured tissues to produce food?
Yes, absolutely. Food is a very emotive topic for people. When people think of food, it's, bringing people together, it's celebrating, it's, it's home for a lot of people, is the food that's served there. So it really gets to the heart of people. So getting people to accept these very, very novel foods is a challenge. And you have people that are at absolute no, I would never eat that. It's, they're totally disgusted by it, to, people that are so willing to try it, to try and, do their part to save the world, I guess, and, to let our oceans recover. For ethical reasons, people want this kind of product. But it's a tricky space to be in. And of course, here in New Zealand, we have an indigenous population here that have this kin-centric ecology with their fish, and we need to, be considerate of their feelings about this technology. And actually, it forms a significant part of the research program, where we look at the perceptions of this technology to both, non-Maori and Maori in New Zealand, and then see what's happening, how New Zealanders feel compared to the global sector. Because in countries like Singapore, they're very positive towards the technology, whereas in some places like Italy and some states in the US, Florida and Alabama, they've actually banned cultured meat already. There's no products, but they've banned it already. So that whole space is a minefield, incredibly interesting. And I love talking to people about cultured meat, because you'll normally get a quick answer one way or the other, whether people are interested or not. And I love just having those conversations with people and seeing, what do you think and why do you feel that way? And in many cases, just being able to give somebody the information about exactly what happens is enough to make them, think, oh, maybe I would try it. So we're really excited to do that work as part of the bid. It will be the first study globally to look at an Indigenous population and how their perceptions of the technology. So we're really excited to get stuck into that, and we've got a fantastic team led by Maui Hudson out of the University of Waikato working on that project. So excited to see where that goes, for sure.
Do you also have any Maori-led research in terms of the actual cell culture, or any involvement there?
Yes, so we have an industry partner down in Christchurch who is going to work with us on developing not cultured meat, but a cell-derived product using a species that his family have worked with for decades and decades. So again, really excited to get stuck into that work. We're just kind of at the early stages of it now, and we're just all dying to get going on it. So there's a lot happening in that space, and I think we are going to lead the charge in working with Indigenous populations around this technology, because for the most part it's not happening globally as far as I can tell. So yeah, we're really keen to get stuck into that.
That sounds like it could be really rewarding as well in terms of the outputs and relationships.
Yeah, definitely. And I think, New Zealand has a big role to play in this technology, and it's not necessarily all of the work that's happening in the lab. There's a lot of other parts to this technology that needs to be ironed out before we're going to see these products at Countdown or whatever. So I think we're just really excited to get started and play our part in that and really get the ball rolling. So yeah.
And do you think this is going to be a major source of nutrition for people in the future?
Ooh, put me on the spot, Julie. Honest answer is I don't know. I think the early promises of the sector have left a lot of people feeling let down and possibly even lied to. And I don't think that was necessarily that, the scientists were being dishonest. I think maybe it was, again, that naivety and optimism that we could do this and we could do it, very quickly. But the challenges, there's still a lot of challenges to be overcome. And with that said, there are 2 places in the world where you may be able to get some cultured chicken, in Singapore and in the States. It's quite hard to get your hands on it. There's, a very small amount produced every week. It's quite expensive and there's waiting lists.
And that tends to be in the form of just nuggets, right?
Yes.
It's not fillets with them. Because you have to align the muscle fibers for that as
exactly,
well. Yeah.
Exactly.
Yeah.
So those first generation products are likely to be fish fingers, chicken nuggets, burgers, that kind of thing. But whether we're going to see them as a huge source of our food in our generation, I'm not convinced on it. If you'd asked me 2 years ago, I would have been probably a little bit more optimistic. But right now, I think there's a bit of work to be done. I think it will be part. I don't know how significant the part is going to be. I would like to see it being a significant part, because I think that technology is a great way of meeting the coming protein deficit that we're going to face. So yeah, I guess it's a watch this space.
And sign me up for some taste testing.
You'll be first, Julie. You'll be first.
Excellent. Thank you so much, Georgina, for joining us today on Scigest.
Thanks, Julie.
And thank you out there for listening. I really hope you enjoyed learning about fish cells with me, and please don't forget to rate and subscribe to Scigest. Mā te ora, until next time.